And it's one of the most interesting topics, so I love finishing the class off with this. It's sort of theoretical, it's a little bit, it's not out there, but it's a different topic than we've considered previously in our diabetes and obesity health-related degrees. This is not something that you often focus on, and it's a good primer for Dr. Kane's aging course next year. So the purpose of this lecture is to see whether we can make a case for exercise in increasing lifespan. Increasing lifespan. We have a few different models we can look at. We have a couple different papers and areas that add pieces to the puzzle, and I'll hopefully build a case over today's class that says perhaps. We still don't know yes or no, but perhaps exercise can influence the lifespan. And our understanding currently about what lifespan is, or what regulates lifespan, is shown in a nice pictograph form here. There are 9 or 10 different hallmarks of aging, things that always seem to go wrong. And so, if these always go wrong, perhaps if we address them all, or one or two of them, maybe we can delay the onset of aging, slow the aging process, or stop aging altogether. And depending on who you ask, the first person that will live to 150 or 200 years old is alive today with our current advances and our rudimentary understanding. As we develop this further, people will start to live a lot longer. Nice to say that. It's not a testable hypothesis yet, and when it is, they'll be dead, but it's nice to say that. That's fine. So we're going to look at 4 different aspects today. We're going to look at genomic instability, mitochondrial dysfunction, the attrition of telomeres, and then inter- and intracellular communication. So signaling factors between cells of the body, hormonal signals, and then factors within the cell. So there's really 5 elements that we're going to look at today. Not all 9, and maybe even not all 5, depending on how long-winded I am today. But really cool information here, so we'll just get started. Genomic instability being number 1. What do we mean by genomic instability? Simply put, DNA damage. DNA is the blueprint in all the cells of the body to make proteins needed for normal function. If that's damaged, the proteins aren't made properly. If they're not made properly, they're shaped differently, and structure drives function. They can't function properly if they're not synthesized in the right way. So we don't want DNA damage to occur. And it doesn't matter what the examples of damage are, whether some bases are removed and we repair the excised or the removed bases, or if some bases are swapped. It doesn't matter what all these acronyms stand for, as long as we understand that some damage on one side favors the aging process and favors a decrease in function, whereas adequate repair on the other favors an increase in longevity. At least that's the hypothesis. It might not be that simple, because it seems as though this whole process is regulated, that it's imposed, and it's not by accident. But if these accidental deficits can be corrected, maybe we have a better shot at living longer. So one really elegant model that looks at this targets specifically mitochondrial DNA. We don't have a good idea of nuclear DNA and its roles yet, but mitochondrial DNA we know are central to apoptosis in the cell. And apoptosis is programmed cell death. When something goes wrong with the mitochondria, when something goes wrong with the genes in the mitochondria, they react poorly and trigger the cell to die, cascades that initiate cell death. This also blends into the next topic, the mitochondrial instability, but we're looking at the genome right now. Mitochondria has its own set of genes. In my younger days, I remembered how many there were. It's either 13 or 31. It's a small complement of genes, but they're housed locally in a circular vector that we know as mitochondrial DNA. And so some researchers have developed a mouse model that lacks the repair mechanism for mitochondrial DNA. This is called polymerase gamma. It's the protein that corrects all those errors that we looked at on the last slide. If it's there, DNA should be intact. If it's not there, lots of errors occur. And so it's a model of accelerated aging. Lots of errors accumulate because this thing has been knocked out. And it's been knocked out in all mitochondria. Liver mitochondria, adipose mitochondria, muscle mitochondria, brain mitochondria, systemic knockout. So if mitochondrial DNA protection is a driver of aging, this would be a good example of accelerated aging. And we can use this model to test our exercise intervention. If exercise is good, as we learned last week, if exercise is good, then maybe there's a potential rescue of function that we can observe. If these mice age quickly and then we have them exercise, what might happen? The comparison that we're looking at are adolescent mice. 4-week-old mice are the equivalent of 12 to 15-year-old children. So into puberty, almost fully developed. Equal groups of normal or wild type. We have the knockout that have just been sitting around and are sedentary, and the knockout that have been undergoing an exercise program their entire life. So we're comparing the ability of exercise to rescue or correct whatever errors occur with this POLG knockout. And I could just show you this picture, not show you any data, and convey the message adequately. This is just a visual comparison of both of the knockout mice. On the left, those that have been exercising. On the right, the sedentary mice. Same knockout. An obvious phenotypic difference. A DNA mutation, the knockout of polymerase gamma, has some effect that's clearly borne out at the organism level. But what is that effect? And maybe this is just looks. Maybe it's like a costume these guys are wearing. Is there actually a deficit in function? Or more specifically, these girls. These are female mice. So let's take a quick look at these mice in action. See if you can guess which is which. These are both knockout mice. 2 of them have been training. 2 of them were the endurance trained mice. 2 of them are sedentary. Both of the same age, and it's pretty obvious which is which. Form drives function, right? These 2 ladies look pretty frail, and they're smaller. Their coats are less lush. They're not moving around very much. They don't seem very active. They're getting crawled over by the more active mice. So exercise does something. Does it completely restore function, or are the wild types, the normal mice, still better off? This is a comparison of a normal wild type mouse versus the endurance trained POLG knockout. See if you can tell which is which. At the surface level, they look pretty similar. Both pretty active, similar size, lush coats, crawling around all over the place. One on the left is the POLG endurance trained mouse. Wild type on the right, POLG endurance trained on the left. Similar function overall. Still inquisitive, still moving around. What is the reason for this response, though? What does exercise do in this model of genetic instability to help rescue function so completely? Well, it protects against mitochondrial DNA errors. This is a plot showing the number of errors in a sample of mitochondrial DNA. You have some in the wild-type mice, like you would expect. You have 2.5 times more in the knockout mice, which you'd expect because they don't have the correction system in place. And then the endurance-trained mice, there's a complete restoration back to normal levels. They've removed the influence of those mitochondrial errors. There's no extra DNA damage in the endurance-trained mice for some reason. And not only that, they live longer. So the mice that are sedentary with this knockout, the large degree of damage decreases their survivability. They live half as long as the other two groups. In fact, it seems as though the endurance trained and the wild type live just as long, 500 days on average. Now, it's a pretty convincing finding, but we don't know if this would be borne out long term. So a mouse would live a lot longer than just 15 weeks, which is where approximately we're making that last measurement. And it looks like the POLG mice are starting to fall off a little bit, so if you continue the measurements, maybe there's some separation. Maybe it's not a complete rescue of function, but still pretty striking considering that the mutant mice live half as long. Pretty potent rescue of function here by the endurance-trained mice. Now, we don't know why this happens. It's probably not that exercise corrects the errors that are there. It's probably more likely that exercise protects against the formation of errors. This is a paradigm where these mice have been exercising their entire lives, and we still don't know what the protective mechanism would be, but it's not likely that exercise would correct errors in DNA. At least I don't know what the mechanism would be. The protein's not there to do it, so I'm not sure how it would execute that task. Anyways, there's a protective effect that we're convinced of. Yeah, Dylan?
So, just like on the slide it says that the final measure is at 15 weeks. Which measure is that?
This is the final measurement here, the lifespan measure. So they stopped at 15 weeks
Okay, just because.
and didn't continue on.
Okay, it just says age 500 days. And then on the slides that you sent us, it says final measure at 1 year, 3 months.
Well, that is one, maybe that's months.
Okay, yeah, okay.
Yeah,
that
500,
makes sense.
okay, that makes much more sense. Yeah,
Yeah.
that's a typo. That should be months.
Okay.
And then 24 months
Yeah.
is the mouse lifespan. Yeah, good catch. Absolutely. Yeah. So error on my slide. I'm going to make a quick note about that. Okay. Not only is this rescue of function seen in mitochondrial DNA and in lifespan, but all of the tissues seem to be improved. We see brain weight, which was compromised, restored to normal. Muscles of the quadriceps, the heart, mass of the ovaries, exhibiting cytochrome oxidase function, which is a mitochondrial enzyme for aerobic respiration, compromised in the POLG mice and almost completely rescued with exercise training. Size of the heart over here. Hypertrophy, cardiac dysfunction in the sedentary mice that's restored with endurance training. So exercise is systemically protective, which is pretty nice. Everything is compromised in the knockouts and rescued near completely in the exercise group. And you can even see at the level of the mitochondria the type of damage that this rampant DNA dysfunction has caused. We have no obvious membrane. Mitochondria are bloated and disrupted. They're not organized. And they're enlarged in the sedentary mice. And we have normal cristae and normal borders of the mitochondria in the wild type that are paralleled by the endurance trained group. They're indistinguishable. I mean, it looks a little bit darker, but the shape and the organization is still there. So whatever the signal is, exercise has this potent ability to protect against the accelerated aging phenotype, at least in this model. But there's a few points that we need to consider. This isn't real life. When you grow mice from pups to young adults to adults with this genetic modification, what other factors are activated or turned off or not working the same as they would in a normal healthy model? There's redundancy all over the place in living organisms. So if this is turned off, what is compensating or trying to compensate? And that's going to affect our interpretation in a free living organism. Either way, we're convinced that in this model, exercise has fantastic restorative effects. We don't know the mechanism, but it protects against all of the deficiencies that we've observed. It's possible we haven't observed all of them. Maybe we didn't make the right measurements. Maybe there are still problems underlying some normal tissue function. But lifespan seems to be protected, and all the other tissues seem to work and be sized appropriately. But it's hard to say if you exercised for your entire life that you would live longer. You can't say that with this data and apply it to humans or even to wild-type mice. And considering that there are 9 or 10 hallmarks of aging, this is really only useful if mitochondrial dysfunction and damage specifically to mitochondrial DNA drive aging. If something else is the main driver of aging, well, we don't know yet if exercise will have as potent effects as shown here. So exercise works in this model. And then applying this model to the population at large or to wild-type mice or humans, that's what we're not sure of yet. That's where the weak link or the link breaks. But it does provide some nice inroads into the activity of mitochondria. So it's not only mitochondrial DNA that might be affected by aging. We've already discussed some of the elements of normal mitochondrial function when we talked about obesity and diabetes. But free radical production may or may not be affected by aging as well. The function of the mitochondria, slippage of electrons. Is there any reason that their function might be impacted by aging? And at an observational level, we can see a pretty wide discrepancy in the free radical reactive oxygen species production between young and elderly humans in this case. 3 times more free radical production, and this is a measure of peroxide production, which is one of our free radicals. It's not the only one, but it's representative and it's easy to measure, so we often use this. 3 times higher production in elderly versus young individuals. And it seems that it's somewhere in the electron transport chain. If we add this compound called rotenone, it blocks electrons from leaving complex 1. So it shuts off the majority of the electron transport chain. Complex 2, 3, 4, ATP synthase, ubiquinone, they don't transport electrons. And so what this is telling us is if we have this massive reduction, there's probably slippage downstream. It's not happening now because we've blocked it with rotenone. So something about the electron transport chain is not functioning properly in the elderly. It produces a lot of peroxide free radicals. But even as we block the rest of the electron transport chain, just complex 1 is showing a bit of slippage. There's still a little difference, even only at complex 1, which is what the rotenone bars are showing us. So overall, we're not sure where it is, but the electron transport chain as a whole seems to not function as well. More electron slippage, more free radical formation.
So then the reactive oxygen species has to come from somewhere before the electron.
This is telling us that it's probably coming from complex 1, and the difference between these is what would be coming from after complex 1. So just seeing an increased production, though, doesn't mean anything insofar as aging. It means these are present and old people age, but there's no cause and effect. There's a theory, though, called the mitochondrial free radical theory of aging. And there's a lot of proponents that are touting this theory that it's production specifically that accelerates mitochondrial damage, that accelerates DNA damage, and then accelerates the aging phenotype as a whole. So if we could ever block or prevent free radical formation, there must be the opportunity to increase lifespan. And according to this theory, if free radicals are the driving factor, that would be the case. If we can block free radical formation, we should not observe aging like we typically do. And one of the candidate organisms that we use, even before we move to mice, is a small roundworm model that lives only about 2 weeks, so really nice for longevity studies. We know the complete genome, we can easily manipulate it, really nice model organism. And if we make these organisms resistant to free radicals, so we reduce the influence of what's there, they live longer. It makes sense in the context of this free radical theory of aging. Less impact of the existing free radicals means longer lifespan. These roundworms aren't mammals, though. We can look in mice, and sort of the flip side of the coin, if these mice are more susceptible to oxidative stress. So like in the first example, we turn off or knock out or knock down certain proteins. Here it's superoxide dismutase, which is one of the primary antioxidants in the cell. If we get rid of that protection, lifespan's shorter. Well, that kind of agrees with the theory. There's more influence of free radicals. There's less protection. And you can see the survival curve shown here. The dotted line are the knockout mice, and they live only 25 instead of 35 months. So if we compromise protection, free radicals can wreak havoc, and they seem to cut our lifespan, or the lifespan of these mice, shorter. But these aren't cause and effect. And there are a couple glaring exceptions to this rule. And a lot of them come from this cute little guy, the naked mole rat, which is a really unique mammal that doesn't live a typical mammalian life, exhibits a high degree of free radical production, yet can live as long as 28 years. Not 28 months, 28 years. That's not a typo, for sure. If you look inside the muscle, they can switch pretty easily between glycolysis and oxidative phosphorylation. They live underground, and I think they can use fructose for energy directly, whereas we as mammals can't. We rely on conversion to glucose. Really interesting metabolism. They produce a ton of free radicals, high levels of oxidative DNA damage, which flies in the face of their 28-year lifespan. This happens at the same time as they live to 28 years. And then, if we want to put the nail in the coffin, we saw on the last slide, okay, if we compromise the antioxidant function in mice, lifespan goes down. According to this theory of aging, if we increase the antioxidant protection, like we did in the C. elegans and the roundworms, we should see an increase in lifespan in mice as well, and that's not the case. So we tried to repeat the experiment in mice here that we did in those small roundworms, where improving antioxidant capacity. Instead of knocking out a gene, we're activating it, we're turning it on, we're overcompensating. And this is superoxide dismutase again, so antioxidant protection, now 1.5 times greater in both young and old mice. No effect on lifespan. According to this theory, if free radicals are the operative factor that causes aging, we remove protection, we age faster. If we add protection, we should age slower, and that's absolutely not the case. Both wild type and these transgenic mice that overexpress superoxide dismutase live or have similar lifespans. So maybe free radicals are permissive. If you have too much, that's a signal something's going wrong. And they can cause damage and cause you to live a shorter life. But they're not the operative factors that can increase lifespan. This has since been fairly well defeated, I think, in the scientific arena. As far as increasing lifespan goes, there isn't much of a role for free radicals, but there is a role for limiting lifespan if they are overly abundant. So we're not convinced that there's much of a role for free radicals in increasing lifespan. There is a role for affecting lifespan, specifically in decreasing lifespan, but not in increasing it. What about telomeres, our third aspect of, our third hallmark of aging? Telomeres are typically what we think of as leading to cell senescence, leading to the languishing of cells in the body. And telomeres specifically are the end caps on chromosomes. The analogy that's often thrown around are the plastic tips on the ends of your shoelaces that allow you to lace your shoes up properly. If they ever wear down, you can't lace your shoes properly. Telomeres are required for normal chromosome division. And every time the chromosome divides, part of the telomere is eroded. It wears away. The proteins that do the translation can't get the entire way, can't replicate the entire strand length of the chromosome. And so these are like those little pins you put in the ends of cobs of corn. They hold on to the genes so that the proteins can do their work. If you don't have those end caps, the machinery that does the replication can't bind, can't do it completely, and so chromosomes can't be replicated. Chromosomes can't be replicated, can't undergo mitosis, cells can't divide, they become senescent. And every cell has a finite telomere length, and therefore there is a finite number of times that cell can divide. It's usually in the range of 30 to 40. And that limit is known as the Hayflick limit for the scientists that observed it for the first time. There's a finite capacity for cell division. It's based on telomere length. When telomeres are eroded and they are too short, cells cannot divide. They become senescent. There's a whole host of other things that happen afterwards. They start to secrete toxic molecules and hormones and cytokines. That's a tangent. They're only able to divide so much. So this could provide the opportunity for therapy. What if exercise or what if a drug could prevent the shortening of telomeres or elongate telomeres? Is the wearing down of telomeres specifically related to aging? Well, it seems if you look in older individuals, well, of course, their cells have divided more. They should have longer telomeres, and we see that. Older cells in older individuals have shorter telomeres. So there's an association present. It seems as though telomere length can be a genetic component transmitted down through generations. and it seems to be somewhat related to longevity, but we'll talk about that in a second. Also, the rate of shortening seems to be proportional to the levels of oxidative stress, bringing in the idea of mitochondrial dysfunction that we just talked about. Perhaps it's not free radicals per se that accelerate aging. Maybe they have a secondary effect on degrading telomeres and accelerating the appearance of senescent cells in the body. Who knows? Maybe there's some interplay. But what if we can prevent this from happening? Does that mean we'd live longer? There is a way to restore telomere length, and there are enzymes within the cell, and they're expressed more in some organisms than in others. Enzymes called telomerases that are specific in elongating telomeres. They don't do anything else but add base pairs to the telomeres and grow the telomeres. So instead of degradation after division, they're restored, and then cells can divide mitotically indefinitely. We don't have it in humans. After we mature, telomerase shuts off, or itself, it's not produced, or it's degraded. Most adult cells do not express telomerase. We have it in egg and sperm cells, and as the embryo develops, but we as humans don't develop it, or we don't exhibit it. Well, that jives with our idea of what might cause aging. So if we don't exhibit it and we get older, maybe if we could institute telomerase, we could increase lifespan. There are some organisms that do exhibit telomerase, and there are cell lines that exhibit telomerase as well. It's a hallmark of all immortal cell lines, which sounds nice because that's what we're reaching towards: immortality, increased lifespan. Let's talk a bit about what these immortal cell lines are. So HeLa cells are named for the lady from which they were first derived, Henrietta Lacks. And these are used all over the place in basic bench science. They have been used to test cosmetics, radiation exposure. They've been sent to outer space. They are proliferative. They are resilient. And they're great for testing extreme treatments. They've been infected with polio to test vaccines. They are so proliferative, they've invaded most other cell cultures. They've been found in laboratories from America to Moscow. They've infected cell cultures that have been transported across the globe, and they can't be killed. Which sounds a lot like cancer, which is cancer. And therein lies the problem of increasing lifespan by activating telomerase. How do you ensure that you only increase normal cell division and not cancerous cell division? I think a lot of people would be much less likely to take a pill or be injected with an enzyme that promotes telomerase when they know, oh, cancer cells, cancer cell lines. The reason they're cancerous is because they exhibit telomerase. It's not a hard and fast rule that if cells exhibit telomerase, you develop cancer. There are some organisms that naturally exhibit telomerase, like the lobster and rainbow trout. These have extremely high levels of telomerase and live, for their size, a very, very long time. Cells continue to divide. There is no cell senescence, or very little cell senescence. And in lobsters, they can live up to 50 some odd years, which is incredibly long for such an organism. I don't know what that would equate to in human years, but probably longer than now. Probably longer than 100.
Is there like a reason that it's just American lobster?
It might just be that those are the only ones that we've looked at. I don't know why it's just American lobster. Not sure. let's just say if there was a way to safely induce this, there's potential here for increasing lifespan. What do we observe in humans? Well, we observe older individuals have shorter telomeres like you would expect. They've undergone more cell division. And so if you look here, these are mononuclear cells and lymphocytes. These are just candidate cells in the immune system. Just like any cell in the body, they have genes that divide, and you can measure telomere length. We see compromised telomere length in the aged controls, at least in lymphocytes. Mononuclear cells don't show that same compromise. They seem to react somewhat differently. But what's important here is that individuals that exercise, or that are considered athletes, don't exhibit the same shortening of telomeres. Or maybe I'll rephrase it. Not shortening. They don't exhibit the same degree of shortness. And that's an important distinction because that implies that athletes don't shorten as quickly, and there's no cause and effect here. This is simply taking young and old individuals that are either control sedentary or athletes and measuring those groups for telomere length. It could simply be that for some reason, if you have longer telomeres and you're older, you're more likely to exercise. We have no idea whether or not it's permissive. We have no idea whether or not exercise made these telomeres longer. But one single 30-minute treadmill run increased telomerase activity in young, healthy adults. There's no way to ascribe this to the aging process or draw this out to lifespan. But if telomere shortening limits cell division, and if that's a driving factor in senescence and mortality, presumably having a higher degree of telomerase activity might delay or prevent that. Yeah?
Going off of what we talked about with the last slide then, couldn't you also argue then that exercise increases the rate of cancer?
You could argue that. You could argue that exercise might increase the likelihood of developing cancer. That's possible. If this is the sole driving factor in initiating cancerous cell lines or developing tumors, this might be a precursor. It's possible. There's no downstream measure, though. Yeah?
On the second graph, is there a significant difference between the young controls and the young athletes, or just the age controls?
Second graph, just young controls and age controls. So the age controls have a shorter telomere length in the lymphocytes. All other groups have similar telomere length. So this is meant to show a rescue of structure, not a rescue of function, a rescue of structure. The older athletes don't have short telomeres like their sedentary counterparts do. We don't know if they lived longer, so we can't draw conclusions about aging. But promising ingredients to the aging recipe here. What other factors might impact aging? This is where I think it gets really interesting. The communication between cells and within the cells, or signaling cascades and hormones, have a large role in growth and development and in initiating the aging program. And specifically, we have pretty good evidence that anabolic hormones are implicated in the aging process. And those are insulin, insulin-like growth factor, and growth hormone. These naturally are produced. The hypothalamus stimulates growth hormone release from the pituitary, and then growth hormone activates growth hormone receptors throughout the body, so in the liver, or in muscle, or in bone, to grow. Right? Growth hormone grows. Growth hormone also stimulates IGF-1 production, which is in and of itself an anabolic type hormone. Both of these in tandem try to increase muscle size, increase hyperplasia, which is the number of muscle cells, so they help with hypertrophy and growing muscle in a weight training program, for instance. Bone remodeling to increase bone mineral density and decrease frailty, to stimulate lipolysis and fat oxidation. These are all what we would consider beneficial effects. And they do support normal function or even improve normal function. But normal function is not lifespan. And so our focus today is lifespan. I also want to point out while I'm here, this will come into play later, growth hormone activates growth hormone receptors. IGF-1, when it's released, activates IGF-1 receptors. And another name for this is called DAF2. These are the exact same thing. DAF2 is the IGF receptor, but DAF2 is in that roundworm model. The IGF-1 receptor in humans is DAF2 in that roundworm. And I'll say that a couple more times. It comes into play at the end when we talk about one big systemic series of events that might affect longevity. Well, anabolic hormones decrease with age. Their release decreases with age, so there's a correlation here. Maybe lower levels of anabolic hormones is implicated in the aging process somehow. Normally, growth hormone is released in high amounts, in large pulses throughout the day in young individuals, and we see that in the blue traces here. Over the course of the day, there are multiple pulses. In the old models, the red squares, there's still some undulation, but much lower overall. Growth hormone release is much lower overall. And if you trace the average value over time, we see this progressive decline in growth hormone, and here specifically IGF-1, in older, this is a mouse model, in older mice. So decreased anabolic hormone release and concentrations with age. Maybe that's why we age. So let's look in a model with compromised growth hormone release. Let's see if we can preserve or restore their lifespan. Except, hold on. These dwarf model mice that have a low growth hormone production, which is what we would see in aging, that we expect limits the lifespan, live longer. They live 40% longer than control animals that have normal growth hormone release. Well, this is completely backwards. If anabolic hormones and their falling off or decrease over time are implicated in the aging process, this is the opposite of that. This says that they have a stimulatory effect on the aging process. And I'm going to get into how growth hormones outside of the cell activate aging inside of the cell in 4.2, which is intracellular factors, or on your slides, metabolic factors, but I thought it would be nicer to connect these two. After growth hormone and IGF-1 reach a target cell, what happens inside that may or may not activate longevity? Anabolic signals almost always signal growth and situations of plenty and abundance, and they activate a protein in the cell called mTOR, which is mammalian target of rapamycin. And mTOR is a protein at the center of a signaling cascade that you often hear talked about with hypertrophy and weight training. It's the muscle growth signaling cascade. We'll leave it at that. So mTOR, mammalian target of rapamycin, the name describes what rapamycin does to it. Rapamycin is a protein that targets mTOR, and it targets and inactivates mTOR. So inside the cell, we have a second piece of evidence, and we're going to try to connect these two. mTOR seems to be involved in regulating lifespan as well. If I ever target it and inactivate mTOR using rapamycin, mice tend to live longer. The survival curves are pushed to the right. More mice are alive at any given point in time. So by turning off that anabolic signal, lifespan seems to be improved. It's a small improvement, but it seems to be improved. Is there ever a way that we can connect the anabolic signals and hormones outside with mTOR inside? Absolutely. And this is a simplified trace of the mTOR pathway. You've seen cascades like this before. You probably know insulin signaling quite well. Maybe you've looked at PGC1 alpha or AMPK as a signaling cascade. There's a whole host of intertwining arrows that activate and deactivate. And what we're looking at here is the connection between anabolic signals in the blood that are circulating, how they activate growth receptors, again, IGF-1 receptor or DAF2, same thing. And this institutes a whole series of signals that eventually activate mTOR. So if, in those Snell dwarfs, for instance, we observe a longer lifespan, well, it seems as though less IGF-1 would probably mean less activation of the insulin growth factor receptor. That means less activation of mTOR and maybe longer lifespan. On the other hand, we just saw that if we add rapamycin, which is shown here, we can turn off mTOR artificially, similar effect to lowering IGF-1 and growth hormone in the circulation. Both of those elements seem to improve lifespan. Overall, the message seems to be that mTOR as a growth or abundance signal, if we turn off that growth or abundance signal, we seem to observe an increase in lifespan. Which, as I say it, logically, it doesn't make a lot of sense, right? You'd expect that growth and plenty would be related to more resources and an increased lifespan. Logically, that would be where my brain would go. It's so backwards that turning off this signal that's involved in protein synthesis, that's involved in gene transcription, that's involved in the organization of muscle fibers and actin, that would reduce lifespan. Yet it does. Yeah?
Can you kind of look at it like that if something's smaller, it needs less metabolic activity to sustain its life?
Yes, and that's how we frame it. So turning off the signal of plenty or abundance seems to concomitantly turn on housekeeping genes,
Okay, yeah.
preservation genes, instead of just using the resources we have because there's so much and being wasteful. The cell turns towards preserving what's already there and minimizing excess waste. And that seems to be the response that improves longevity.
So it's not necessarily that you're increasing lifespan with the same level of like life, you know what I mean? Like
I'm not sure what you
I'm
mean.
not sure how to phrase it. Like because you're reducing the metabolic activity, you wouldn't necessarily be the same as you were when you were younger. Like, you know what I mean? Like if you were, let's say, able to run a 5-minute mile, just because you increase your lifespan, you wouldn't be able to run the 5-minute mile longer. It would decrease.
I sort of follow,
Yeah,
but not quite. We're going to go into an example that does get at this from not an exercise point of view and not a hormone point of view, but I think is a really fascinating example. And let's see if that kind of brings this this point back around. We also have, I think I'll show you the evidence. Yeah, if we reduce growth hormone, live longer. If we turn off mTOR, live longer. if we block this receptor, live longer. So turning off DAF2 in those little roundworms, you live longer. Overall, reducing that situation of plenty seems to increase lifespan. Yeah, Peter?
Because it's a response to like the idea of abundance, would like diet have an impact into this?
Yes, absolutely. Diet would have a large impact into this. Let's get there. I threw this next slide up to show you that, yeah, it seems complicated on the last figure, but this is mTOR, and this is mTOR1. So there's mTOR1 and mTOR C2, and you've got insulin up here, and there's a whole host of other signaling factors, and it gets a lot more complicated, so only know the other one, but this shows you the level of complexity within the cell and how it reaches into other pathways and is involved in other responses. But to Peter's point, diet can play a large part in the hormonal response and ultimately lifespan. And I find this to be a fascinating example. The example of intermittent fasting, temporary starvation, or calorie restriction. This is one of the most proven, robust ways that we have to increase lifespan in all organisms. Roundworms, mice, monkeys. Don't know about humans yet because we haven't done that study. Can't really do it too well. Very robust, conserved response. Conserved response to increase longevity in many unrelated species. And the fact that we see it in all the model organisms as well as closely related mammals means it's important. Means it's a shared response that provides some key benefit regardless of the individual variations in the organism specifically. There's a universal benefit to those organisms. Hence, it's conserved. The question then becomes, why? Why would it ever be a benefit? And why would it be a benefit across so many various organisms? Why would the absence of food, the absence of energy, prolong lifespan? Let's walk through an example. Certainly in the extremes, I'll say this right out. In the extremes, it's not beneficial to lifespan. Starvation will kill you. In extremes, it's detrimental. But the restriction of calories we often think of as a 30 to 40% reduction in energy intake. So let's say MyFitnessPal tells you to eat 2,000 kcals per day. All right? 30% decrease is 600 kcals less per day, so you're eating instead 1,400 kcals per day. That's still very little food. You could survive on it. It's not starvation. That's the kind of calorie restriction that is this conserved response to increased longevity. So why would this ever be a benefit? You have to imagine in a past life, in previous years when abundance wasn't likely, when we didn't have easy access to food or drugs, we didn't have cushy desk jobs, and we were hunter-gatherers, we would often run into famine. Feeding cycles were cyclical. You'd have a big hunt, eat a mammoth, be great, and then a couple months later, mammoth is gone, nothing to eat. Periods of famine were rampant in the not-so-distant past. And what this tells us, just in theory, there's no food around, it's a bad time to reproduce. Unfortunately, very little food means less likelihood that your offspring survive. So, we need a way to be able to wait. We need a way to survive long enough to delay reproduction until we find that next mammoth, or we enter a period of abundance or surplus, or we find food, until famine's done. This hypothetical way to increase lifespan and improve reproduction then would allow more reproductive success. I'm not going to reproduce in periods of famine because most of my offspring will die. Instead, I'm going to wait until a period where food is plentiful or abundant, and maybe some offspring won't make it, but the large majority will. And that's really the whole point of life as we know it, passing on your genes to the next generation. As a very utilitarian process, life is division, replication, and growth. And chasing your passion. But this is the schematic for calorie restriction. And it turns out that the increase in lifespan is only a secondary effect. The whole purpose of staying alive longer is to delay reproduction. And we're not going to look at the evidence here, but there's really interesting evidence from a guy named Jay Olshansky in, I forget where he is, in the States, and I want to say Illinois or Connecticut, that ties hormone cycles and release in puberty and during reproduction to the lifespan, to the overall window that you are alive. And if you delay reproduction, it pushes the lifespan window back. It's the ability to delay reproduction that makes you live longer. The reasons are hormonal and we don't fully understand them, but the concept makes sense, especially for us, because as humans we have pretty useless babies, right? 18 years you need to invest in them, and sometimes longer before they're self-sufficient. Deers, horses, mice, cats, maybe a couple years, but you still need to foster and nurture them as they grow. So pushing back reproduction necessarily increases the lifespan. We think that the signal is growth hormone and IGF-1 related, which has interesting implications for our current situation of abundance. If you think about growth hormone release, insulin like growth factor always being released in response to plentiful meals and being sedentary, there's probably some pretty bad downside that will be borne out shortly for our sedentary society. We think growth hormone and IGF-1 are the operative factors, and we see a nice progressive stepwise improvement in lifespan by modifying the cell's ability to respond to growth hormone. So this is why I brought up that DAF2 connection. DAF2, which is the IGF receptor in C. elegans. If you knock it out, you increase lifespan. Here, normal roundworms live 35 or 40 days. If you calorie restrict those roundworms, so you put them in a medium with less sugar than they want, they live longer, but not much longer. And that's, we think, due to some of the changes in the housekeeping genes in the cell, and there's a whole host of other work by Cynthia Kenyon out of Stanford, and this is a paper from her lab that we're not going to talk about. But if you calorie restrict, they live longer. If you knock out the IGF receptor, they live almost twice as long. And then if you add the two together, the response is even more beneficial. So no influence of growth hormone, plus you have the calorie restrictive housekeeping effects within the cell. There's a nice stepwise additive progression here where the overall lifespan is doubled in these roundworms. Lifespan doesn't double for all organisms, but it is still conserved. Lifespan in mice and in primates seems to increase about 30 or 40%. Not 100%, but 30 or 40% is still pretty good. If we live to 100 and we get that response, we're now living to 130, 140. The sacrifice is you live on 1,400 out of 2,000 kcals per day every day of your life. Is that living? Therein lies the distinction between lifespan and health span, and that's the topic for another course. But it's a conserved response that you can see borne out here. These are rhesus macaques, and this is a paper that leads into one of the ones we're discussing on Thursday, One of the ones on Thursday takes this study and draws it out longer term. So in 2009, I think these monkeys were 12 to 13 years old. One group calorie restricted, one living normally. Older normal monkeys, calorie restricted monkeys. Just by appearance and posture, you can tell there's some benefit to calorie restriction. The longer term study looks to see about lifespan and health span and if this response actually is borne out in monkeys, and hoping to ascribe that to humans as well. This sets the stage for a new and cutting edge potential therapy for aging. And I really shortened the discussion on this part because we've always run out of time when I've tried to present it before. There's a guy named Nir Barzilay at the Albert Einstein Institute of Medicine in New York that is really pioneering this work. And this was his proposal only 2 years ago, and this is undergoing clinical trials and hypothesis testing now. We don't have results of this, but metformin, a common anti-diabetes drug, has pretty significant potential to increase lifespan in humans as well. And the idea stemmed from the fact that in their trials, treating diabetes and observing people long term, the treatment group always tended to live longer. And for the longest time, they ascribed that to, oh, well, diabetes has been reduced or insulin sensitivity has been restored. But when looking at the mechanisms of this diabetes medication and how it can impact the cell, it became clear there are a lot of potential inroads to increasing lifespan apart from improving diabetes. And we've since characterized many different inroads of metformin. Metformin inactivates the IGF receptor directly. Just like calorie restriction decreases growth hormone, or the Snell dwarfs had lower circulating growth hormone, or if you knock out DAF2, metformin does that. That's a pretty, if that's a conserved response that increases lifespan, that's a nice effect of metformin. What else have we talked about today? mTOR. Metformin in the cell activates something that turns off mTOR. So in short, metformin turns off mTOR. Well, we saw today that if you turn off that signal of abundance and surplus, lifespan is universally increased to varying degrees, but it's increased. Metformin does that. We didn't look today at inflammation, but there's a lot of there's a growing body of research that characterizes obesity as a systemic low-grade inflammation. And these inflammatory cytokines like PI1 or TNF-alpha you've heard about, IL-6, there's a whole host of them. They're released from adipose tissue and generally induce cell death and cellular dysregulation. Metformin reduces inflammation. So there are inroads for those researchers on that side of the coin. We also have some of the genes involved in housekeeping. We didn't talk about this in depth, but that housekeeping response is called autophagy. It's called autophagy. And autophagy is accentuated when mTOR is turned off. So as a consequence of reducing that signal of abundance, it removes the brakes, so to speak. It removes the brakes on autophagy, and autophagy helps to increase health span and longevity. Multifaceted, multidimensional impact of metformin on potential factors involved in the aging process. And so this has a lot of promise for aging research, and these results will hopefully be available soon, but this is ongoing, cutting-edge stuff. You'll also probably like to know about this area over here, resveratrol. You've heard of resveratrol? The compound in the skin of red grapes. That's typically abundant in red wine. That's supposed to confer health benefits. That's why you have a glass of red wine a day. Resveratrol, also implicated in health span and lifespan, activating what are called the sirtuins, which eventually increase cell survival, we'll put it that way. Metformin's not implicated necessarily in this response, but red wine certainly is. Although I won't tell you that you need to drink 36 bottles a day to get the benefit that we see in the pharmacological studies. There is some suggestion that resveratrol can improve health span and longevity through that adjunctive pathway. So good news. That's the take home for today, right? Yeah.
Well, alcoholics live longer, you're saying.
Only
Only red wine.
Moderate alcoholism may or may not impact lifespan. I've also seen a few articles come out recently that apparently no amount of alcohol on a daily basis is good for you, and these trace amounts of resveratrol are nothing. They pale in comparison to the amounts that you need to induce these effects. So I'll leave it to you to do your own research by reading primary articles, not any other experimentation. All right, let's summarize. There have been a lot of different points flying around, and if we can bring it all together, we've gone away from exercise a little bit as far as impacting longevity and lifespan. But I want to articulate first that it's unlikely there's one thing that's responsible for aging. It's unlikely that it's just growth hormone, or that it's just mitochondrial dysfunction or telomeres. Yeah,
I just have a question about caloric restriction.
sure.
So has there been any studies done where they reduce the net caloric intake by exercising rather than just restricting food intake?
Yeah. So there are a lot of really good studies on the pattern of caloric restriction, which, like you mentioned, Odessa, is either exercise or not eating as much, or changing the way that you calorie restrict. So there are paradigms now where you alternate day fasting, eat one day, don't eat the next, so on and so forth, or eat for 2 days, don't eat for the third day, and you repeat that, or you just don't eat from 6 p.m. or 4 p.m. onwards. As far as the deficit created by exercise, I don't know how those are comparable directly, but looking back to the obesity section, remember we compared those directly in obesity, and both had really nice stimulatory effects on insulin sensitivity. They both reduced body fat and improved body composition. I have to believe that there's some benefit. Actually, I'm talking myself out of it as I say it. So my first thought is there's some benefit to having the extra metabolic machinery. The cellular fitness probably should provide some benefit as far as longevity is concerned. But then, if we consider that upkeep of that cellular machinery might impact longevity, there's more mitochondria around, there's more protein around that needs more energy to maintain. Maybe that's a detriment long term. I don't know how that's going to be borne out, but I don't think they've compared directly those two. It's an interesting prospect, though. Especially considering that the candidate model is often C. elegans. It's hard to get roundworms to exercise. But with mice, you could probably do it. I don't know if that's been compared directly yet. Good point, though. So no one thing causes aging. I think it's a program. There are many different factors, kind of like there's a lot of factors that result in the accumulation of energy and obesity and diabetes. A lot of factors contribute to aging. If one of those factors is genomic instability, specifically damage to the mitochondrial DNA, we saw exercise as a potent therapy to recover and reverse those effects. There's no way to say that it would be as severe in a free-living wild-type human or mouse. And it's difficult to say that one bout of exercise would have the same magnitude of benefit as a lifelong exercise program, but exercise can restore the damage done in those POL-G mice. We looked at free radical formation. It could be that genetic damage in the mitochondria leads to an accelerated free radical production. And there does seem to be some effect whereby an accelerated or increased production of free radicals might shorten lifespan. But it doesn't seem that reducing that impact will increase lifespan. So it doesn't seem to be the only factor involved in lifespan. If this were the main driver, then reducing this impact would increase lifespan always. Something else seems to be regulating the aging process on top of free radical production. And that might be telomeres. This is a physical limitation of all the cells. We articulated that this might even be impacted by oxidative stress, that the wearing down of telomeres might be increased or accelerated with an oxidative, stressful environment. And while there's some promise here that exercise as a therapy can enhance telomere length, we don't know if that enhancement means longer lifespan or means development of cancer. There's not enough evidence either way to jump into a program to try to live longer or to avoid it to avoid cancer. I think that that's a pretty tenuous and foolish correlation, but it's debatable that it will increase lifespan. I think that the master regulators are probably systemic. So far, most of these factors are local, within one cell. Mitochondria in one cell. Damage to the DNA in one cell. But I think the modulating factors are more likely to be systemic. Hormones, growth factors like IGF-1 and growth hormones specifically, and how they influence the cascades within cells across the body. That would be the way, if I were designing it, to institute a universal program to cause accelerated aging. And that would be a really nice starting point to investigate delaying the aging process. And some of our rudimentary attempts have shown just that. So calorie restriction targets this pathway, growth factor and the signaling cascades. Knocking out receptors disrupts the communication of that pathway. And universally longer lifespan when we do that. Getting that into a usable form, though, is what's tricky for humans, because you can't just knock out insulin-like growth factor 1 receptors in humans. You need an analog or something to compete with those receptors, or something to do some of the work in the cell like resveratrol or metformin, or a way that doesn't hurt all the time to calorie restrict. Right? Do you want to live your life hungry? Maybe, maybe not. Depends. So that's the end of this section. I thought this was a pretty interesting spin on some of the material we've discussed so far. We'll leave it there, and we're going to pick up with 2 papers on Thursday that help expand our view of this material. I forget which papers they are, but they build on some of the concepts that we talked about today. Actually, I think one of them is the metformin paper, which could be good. Any questions before we call it for the day? No?